Hello, good afternoon, everyone. We're going to finish lecture 2 today. We still have 3 more macromolecules to talk about, lipids, proteins, and nucleic acid. And I just want to start with a quick note to give you an overview of what to expect in lipids, because lipids is a little bit different compared to the other 3 kinds of macromolecules. By now, you should know macromolecules like carbohydrates consist of multiple repeating units that are linked together using dehydration synthesis, and it can become very, very long. And that's the same situation for proteins and for nucleic acid. However, when it comes to lipids are not really polymers in the sense that it's not consist of repeating unit that goes on for some time. Rather, lipids are molecules that use certain components to create just a big molecule. So because it's a little bit confusing, I think we're gonna go ahead and give you a summary first, and then we're gonna go through the PowerPoint slides. Okay, so here we go. Let's put lipid here in the middle. Lipid. There are different kinds of lipids, and when I say the word lipid, what comes to your mind? What is an example of a lipid that might come to your mind? Yeah, fat. That's a good one, right? So the fancy word for fat is triglyceride. Triglyceride. Okay? It's just the same meaning as fat. Now, how do you make a fat molecule? Well, fat molecule has 2 components, 2 ingredients, if you would. Okay? We have this thing here called a glycerol, and you're gonna need 3 fatty acids. Okay, I'll tell you what these are later. Okay, this is just an overview, okay? So these here, those are fatty acids. And that's it, that's a fat molecule. You can see it's different from carbohydrates in the sense that, take cellulose, for example. Cellulose is a long polysaccharide, consists of glucose connected to glucose to glucose, glucose like hundreds of times, right? But in lipid, it's not like that. You don't have the same unit repeated over and over again. Instead, we have components. We have glycerol, and then we stick 3 fatty acids onto it, and then we get a fat. Now, we might as well summarize the functions here. The function of fat would be storage of energy. Fat is also useful for insulating. And cushioning organs. So it keeps them warm and it gives a little bit of protection. So like when you fall down, there's a little bit of cushioning that protects your liver. So like you don't like burst them or something like that. And there's a little bit of fat around kidneys, right? Around heart. All major organs would have a little bit of fat around them to keep them warm and to protect them. So that is the first kind, right? Triglyceride. All right. Another kind of lipids, they are called phospholipid. Phospholipid. So how do you make a phospholipid? Well, it turns out you also need to have a glycerol. And this time, you only need 2 fatty acids. And one more thing, you are going to need to have a phosphate group. Okay. Do you guys learn phosphate in chemistry yet? Anybody know the formula for phosphate? Not yet?
pH4?
Yeah, pH 4. 3 negative, right? If you didn't know that, that's okay. Well, I'll talk about it later. So what is the function of phospholipid? Well, it is the major component of the cell membrane. Cell membrane is the thing that surrounds your cell and is primarily made up of phospholipids. All right, another kind of lipids would be steroid. So steroids, an example of what it does is it forms hormones. Not all hormones are steroids. Some are actually proteins. But like the sex hormones, those are steroids. Does anybody know the name of the sex hormone for like female, for example?
So estrogen, progesterone, testosterone.
Exactly, you got it, right? Progesterone, estrogen, those are female hormones primarily, and then testosterone is found primarily in male. Those are steroids. And how do you make a steroid? Well, to make a steroid, you need to have these carbon rings, okay? And it doesn't really matter how you draw them. But you will have 4 carbon rings. 4 carbon rings. Now, attached to the carbon rings could be a bunch of other stuff. Okay, like I'm just gonna randomly draw some stuff there. These random things that you stick on them, we call them functional groups. Functional groups. Depending on what kind of functional groups you have on the steroid, it's going to give it different properties and different characteristics. We don't have to learn all the functional groups. Okay. Just know that a steroid has 4 carbon rings and some stuff, some accessory, if you would, that are attached to it.
So excuse me.
Mm-hmm.
So you said what is the difference between the hormones? Just is the number of carbon rings?
Carbon ring is always 4, but what makes them different is the kind of functional groups that they have. Okay, so functional groups are like these special groups of
Oh,
molecules that are
okay.
attached to it, right? Yeah,
Okay, I got it.
good, good, good. And last but not least, we will also look at wax. Okay, wax is important for forming water repelling surface. So if you think of like the skin of an apple, it's waxy. It protects the fruit from moisture. You have waxy stuff on your skin, in your hair. Like when we soak our hand in water, it's not gonna go into it, right? It's gonna stay outside, at least for a while. And that basically is wax. And wax are like fatty acids. Okay, fatty acid plus some. I mean, again, like I'm oversimplifying it for you, but it has a bunch of OHs on it. Okay, and these OHs, you learn it next semester in chemistry. Whenever you have OH attached onto something, um, we call them alcohol. Yeah, so when I say alcohol, like drinking alcohol is only one type of alcohol, right? there are different kinds of alcohol. Drinking alcohol is ethanol, rubbing alcohol is propanol, right? So you can have alcohol, OH, attached onto a fatty acid and you get wax. So that's a quick overview of lipids. And I hope you understand why it is different compared to the three other types of macromolecules, right? Namely, it's not the same unit repeated multiple times. Does anybody have any question before I go to the PowerPoint slide? Already. So lipids. Chemistry of lipids. It surprisingly or not, depends how you look at it, it also contains the same three atoms as carbohydrates, right? C, H, and O, carbon, hydrogen, and oxygen. And there will also be traces of phosphorus for some type. Without looking at your notes, do you remember the four types? Which of the four types has phosphorus in it? Well, the phospholipid is the one that has a phosphate group. So this is the one that has phosphorus in it, okay? The other stuff, the triglyceride, the wax, and the steroid, they are just going to have CH and O. Unlike other macromolecules, lipids are built with components rather than a long chain of monomers. I explained that already. And all lipids are hydrophobic, meaning they cannot dissolve in water. So let's start with fats. Okay? Another name for fats is triglyceride. And as I showed you in the overview note, it's made up of one glycerol and three fatty acids. The function is to store energy and to provide insulation. Fat is excellent for storing energy. The same mass of fat compared to like sugar can store up to 8 times more energy. So how do you make the fat? Well, this is the glycerol molecule. Some people call it the ho molecule. Just make it easier for you to remember, right? Because it has the OH, OH, and the OH here, all connected to a C, C, and C. And then we have fatty acids, right? So fatty acids are basically long chains of C and H. Right, so you can see here is C, 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 and then with a bunch of H. At one end, you have this C double bond O and then OH. But the fatty acids can be long or it can be short. These are quite short, but it's not uncommon for fatty acids to have like 12, 16 carbons in them. They just keep it short here to fit into the diagram, basically. And also, the fatty acids might have only single bonds here. Okay? Single, like this here is called a single bond. There's only sharing of 2 electrons, but some of the fatty acids might have a double bond, right? So this thing here is called a double bond. And that means 4 electrons are being shared. Okay, I know it's this is very chemistry looking, and you will learn about single bond and double bond. But for now, I'm just gonna keep it as simple as possible. Single bond is a single line, double bond is 2 lines, and single bond means 2 electrons are being shared. All right. So how do you connect the fatty acids with the glycerol? Well, it's the same thing. It's a dehydration synthesis, right? We're going to remove the hydrogen, and we're going to remove the OH here, and then you're going to form a connection, and you have to connect 3 fatty acids to the glycerol. That's why you end up having 3 water molecules. Food source, where can you get fat? Well, they are kind of everywhere. If you eat meat, there's going to be some fat in it. And even if you don't eat meat, a lot of the plant products, they are also high in fat. Avocado, for example, has quite a bit of fat. People use coconut oil. Walnuts is good in fat, right? So there's no shortage of fat in our diet in general. All right. There are 2 kinds of fat. There's saturated fat, and then there are unsaturated fat. Okay. Typically, saturated fat is more unhealthy than unsaturated fat. Okay? You want to avoid food that are high in saturated fat, and food that are high that has more unsaturated fat. Like, I mean, you can have more of it, but like I wouldn't overindulge either. So let's take a look at what the difference between the two are. So the saturated fat is made up of glycerol and 3 saturated fatty acids. Saturated fatty acids are fatty acids that only have single bonds in them. Okay, only contain single bond. So all 3 fatty acids have to be saturated in order for you to call the whole fat saturated. Let me just go back here to this picture. All right. Is the first one a saturated fatty acid or an unsaturated fatty acid? I mean, you don't have to answer me. You can think about it, right?
Saturated
Yeah,
because
this
it's two electron, single.
exactly, because it's all single bond, right, in the C and H. So this is saturated. Next one is also saturated. And then the last one has a double bond, so it is
Unsaturated.
Yeah, unsaturated. Okay, so the whole fat, right? Because you have one strand that are unsaturated, then this whole thing is classified as unsaturated fat. Okay, so the second one, no, the second one is saturated right here. This is saturated because all the carbon and hydrogen are single bonds, right? Okay, we don't count the thing at the front. Okay, it's just like within the chain. Okay. So for it to be a saturated fat, all 3 fatty acids have to be saturated, meaning they only have single bond and the carbons are connected to a maximum number of hydrogens. Okay? Carbon can only form 4 bonds. There are, in the case of saturated fat, right? Each carbon is connected to 4 things. 1, 2, 3, 4, right? There's no more room for more hydrogen. And so the structure of saturated fat is kind of like this. Okay, you have glycerol here, and then you have the 3 saturated fatty acids. And the fatty acids are pretty much arranged in like a straight line. It goes zigzag a little bit, but for the most part, it's straight. And that means if I have another fat molecule, you can put it pretty close to each other. Okay, maybe I'll have another one here, right? And they tend to be able to pack together quite nicely. That's why they are mostly solid at room temperature. If molecules are really condensed, really packed together, they tend to be solid at room temperature. And the reason why it is relatively unhealthy compared to unsaturated fat is that they tend to pack closely inside the body as well. So if you have a lot of saturated fat in your diet, then they might start to clot your arteries, and that's not a good thing. Most animal-derived fats are saturated. If you think about butter, lard, right, bacon grease, all those things are saturated fat, and we should consume them in moderation. Unsaturated fat, on the other hand, consists of glycerol and at least 1 unsaturated fatty acid. Okay, at least one unsaturated fatty acid. You can have 2 unsaturated fatty acid. You can have 3 unsaturated fatty acid. Okay, so if fat has 2 or 3 unsaturated fatty acid, then we say polyunsaturated fat. Polyunsaturated fat. The more unsaturated fatty acid it has, relatively speaking, the healthier it is going to be. So what is the difference between unsaturated and saturated? Well, I kind of told you already, right? Unsaturated fatty acid has a double bond. So if you look at the double bond, I'll draw it for you so I don't have to go back. If you have a double bond like that, then actually the carbon has more room, right? You can actually break the double bond and put more hydrogen on it, right? So when there's a double bond, it's not attached to a maximum number of hydrogen. So how does the double bond affect the structure of the fatty acid? Well, anytime, so this one is saturated, this one is saturated, and let's say the last one here is unsaturated. So anytime it has like a double bond, it causes the fatty acid to create a sharp turn, a bend. Okay, so if I have another fatty acid here, okay, and then it has like this sharp bend, then they are not able to pack closely together. It's like if I asked you guys to line up, side by side, if you keep your hands and legs straight down, you can line up closely to each other, right? But if you stick your arm out, much like the double bond here, then the person is not going to be able to get very close to you, okay? And so since they can't pack very nicely together, they are liquid at room temperature. Most plant-derived fats are unsaturated, okay? So give me some example of plant oil that you guys use for cooking. Anybody?
Sunflower.
Sure, sunflower oil, right? Any
Avocado oil.
avocado oil,
Olive oil?
olive
Olive.
oil, yeah, excellent, right? Grapeseed oil, right? All these things are unsaturated fat, okay? Now, remember I told you before, there are always exceptions, right? It's not always this or that. There are some exceptions, right? So coconut oil, it's plant derived, but if you ever use coconut oil, it's kind of like a white paste, right? So it's kind of solid, even though it's plant derived. All right. Now there are some essential fatty acids. Essential fatty acids. The word essential means we cannot make it in the body, but we need them. So the only way to get them is through our diet. Right, so there, the example of essential fatty acid would be omega-3. Okay, I'm sure you've heard of omega-3, and what's the other one? Omega. Have you guys heard of this before? Omega. It's usually on eggs, right? Omega-3, and then there are some other numbers. No, this is the first time you hear about it. 3, 6, 9. Okay, so I'll tell you what those are in a bit. Okay.
6.
Yeah, 6 and 9, exactly. So omega-3, such as something called EPA and DHA, these are essential fatty acids that are recommended by doctors for pregnant women, okay? Because EAP and DHA are important lipids for fetal brain development, okay, for the baby. So that's why pregnant women sometimes would ask to, take these supplements. You can get these essential fatty acids from natural sources, like fish is really rich in them. A lot of the nuts and avocado, it's quite rich in omega-3. Of course, eggs as well. So they're both unsaturated fat, meaning they have double bond in their tail. And depending on where you find the first double bond, that's how they name it, omega-3, 6, or 9. Okay, so over here, this is carbon number 1, 2, 3. The first double bond occur at carbon number 3. Same thing here. So these things are example of omega-3. Okay, you don't have to know this. I'm just telling you in case you're wondering like what exactly does it mean, right? So omega-6, you will find the double bond for the first time on carbon number 6. Right? Okay, so here is a picture that shows you the difference between saturated and unsaturated. And once again, you see the glycerol in both of them. And the glycerol is attached to 3 fatty acids in each case. For the saturated case, no double bond, so all the tails are pretty straight. This one is a polyunsaturated because all 3 tails have double bonds in it, right? In some cases, there are multiple double bonds, and it causes the tails to have these sharp bands or kinks in them. So these would be liquid at room temperature. These would be generally solid at room temperature. Any questions?
So just to clarify, saturated, they have no double bonds,
Good.
and unsaturated has double bonds.
That's right. 1 or more double bond in the tail.
Okay.
Great. Anyone else?
Saturated fat is unhealthy because they are closely packed in a body, that's why.
That's right. So they're close. They generally pack closely together. So even after you consume it, they may pack closely in your artery and clot them, right?
Okay.
Okay. Saturated is bad, but not as bad as trans fat. Okay. What is trans fat? Well, when you take unsaturated fat, which is kind of like the healthier option, right? But because they are like liquid at room temperature, like it's kind of weird to put them on your toast in the morning, right? Like you can like drizzle, I don't know, avocado oil onto your toast and eat it. It's kind of weird, right? So people came up with a way to convert them into spreadable form. And you can take unsaturated fat and you can add hydrogen to it and convert them into saturated fat. This process is called hydrogenation, and that is how you create shortening and margarine. Right? So margarine is actually worse than butter because it's highly processed. It has undergone this hydrogenation process, and in doing the hydrogenation process, it generates a bit of a byproduct called trans fat. Okay? Trans fat cannot be broken down by the body. It just stays in your body for a very, very long time, right? And it's linked to increased risk of heart disease and diabetes. Basically, is really, really bad. Okay? But trans fat has been used in many processed food and fast food because they actually extend shelf life and they make the food taste better, apparently. That's why eating a lot of processed food and fast food usually also increases the risk of heart disease and diabetes. But because it's so bad, Canada actually banned artificial trans fat in 2018. Right? So now people are not allowed to use it to prepare food. However, if they are already there in the food, for example, like in the margarine, because it's a byproduct as part of the process of creating margarine, then it's okay. But like they ban it for being used as an additive on purpose. Okay? You can read more about it at the link there. Okay, that was fat, which is triglyceride. Now we're going to talk about phospholipids. Okay. Phospholipid, as I told you earlier, is a key component of cell membranes. All cell membranes have phospholipids in them. And it's made up of a phosphate head, which is right here. And the phosphate, if you don't already know this, phosphate is PO4 with a charge of -3, okay? TO4 -3. So the head is polar because of a charge, but the tails are going to be non-polar. And that makes it a very special molecule because it has a polar portion and then it has a non-polar portion. Right? You actually should be, I assume, should be familiar with something like this. Every time you use detergent to wash greasy dishes, the detergent is basically something that looks like a phospholipid. It has a polar head group that would attach to the water, and then it has the non-polar tails that would attach to the grease, right? If you just try to wash the oily plate with water alone, it will stay greasy, right? But you add the detergent, it's like a binding agent. It allows the lipid to bind to the water and you can wash it off the plate. Right? So basically phospholipid is similar in that regard. So in addition to CH and O, it also contains P in the head. Okay, so most people would draw the phospholipid like that. Right? That's one of the fatty acid tail. The other fatty acid tail might also be saturated, or in some cases, one of them is going to be unsaturated. Okay, but regardless, you have to know the head is going to be hydrophilic and the tails are going to be hydrophobic. I'll give you a moment to catch up and copy whatever you need. So if you take a look at the cell membrane, okay, this is a cell that has been cut open, and this out here that surrounds the cell, that would be the cell membrane. And if you zoom in, you will see the cell membrane is actually made up of 2 layers of phospholipids. That's one layer, and that's another layer. And you have the hydrophilic heads that are facing outside of the cell, or it's facing inside of the cell. Outside of cell. Because surrounding the cell is mostly water, and the inside of the cell is also mostly water. But the tails are going to be sandwiched between the 2 hydrophilic layers. Okay? So think of this as like a peanut butter sandwich. Right? That's the bread, and then you have a thick layer of peanut butter in the middle. That's basically how the phospholipids form the cell membrane. Okay? We will talk more about cell membrane later on. I think lecture 4. Okay? So this is just a quick overview. Okay, that's it for phospholipids. Right? We talk about triglyceride, phospholipid now. We're going to talk about WEX. Just going to keep it very simple. It covers exposed surfaces of plants, not just the apple, right, but the leaves as well. It protects and lubricates our skin and hair. You can also find wax in things like honeycomb. People can buy like bee wax. What do you call those things that you put on your lips? Chapsticks, right? Okay, lip balm, right? That's basically honeycomb wax. It consists of long fatty acids connected to alcohol. Just want to remind you, alcohol is just O and H. That's basically what it is. That's it for wax. Steroids contains fused carbon rings, 4 of them. So you can see these hexagons, they are basically carbon rings. And it contains various functional groups. So that's a functional group, mix it and alcohol again. There is another functional group here. And cholesterol is actually an important steroid in the body. Okay? You will find cholesterol also in cell membrane, along with phospholipids. And cholesterol are useful because they are starting material for many oops, for many hormones. All right, so if you need to make the testosterone, if you need to make the estrogen and progesterone, the raw material that you start with would be the cholesterol. Okay. Now you might hear people say, "Oh, I have high cholesterol. I need to take medication," or like you need to watch out for the cholesterol, right? It's true, okay? But there are like good and bad cholesterol, right? So not all cholesterol are unhealthy necessarily. Okay? We'll learn more about that next semester. I didn't even know I wrote it down already. So sorry, you wrote down something for nothing. So here it is. That's what estrogen looks like, and you can see, right? You got the rings here, 4 rings, but the functional group that you have attached to the rings, they are quite different. And that's it. That's it for lipids. All right. Let's do poll question. Okay, I'll end the poll. 3 fatty acids and glycerol, that would be triglyceride. Okay? Tri means 3, right? Like a triangle has 3 sides. A tricycle has 3 wheels. Triglyceride has 3 fatty acids. That's how you can remember it. Okay. Phospholipid is glycerol plus 2 fatty acids and a phosphate head. Okay, here's another one. Okay. Steroid has at least 1 fatty acid tail on it. That actually is the incorrect one. Okay. The only thing that steroid needs to have would be 4 carbon rings. Okay, and then you attach these functional groups on them. So it doesn't necessarily have to have a fatty acid tail. So that's the wrong one. Okay. A lot of people chose D as the false one. Let's go back here. Cholesterol is an example of a steroid. In fact, cholesterol is the starting material to make more steroids, okay? Like estrogen and testosterone. Okay, that's it for lipids. Seriously, any questions before we move on to proteins? Protein is like the hardest one. Right? It's, want to make sure you're okay before we move on. I need to get some props. Just give me a second. But you know, now is a good time to ask me anything. Okay, nothing? By the way, your first quiz is gonna be next week, right? Next Thursday. I will post the information about the quiz probably today. It's gonna be on lecture 1 and lecture 2. When I post the information, I'll tell you how many questions to expect. If you have not already done so, please try the self-quiz. That will help you get used to the online testing platform. And you also have to install the lockdown browser in order to do the quiz, right? We do have class on the day of the quiz, okay? So make sure you plan to attend. The quiz will be available from 6 a.m. all the way to 11:59 p.m. At night. So you would have plenty of time to access it. Okay. Proteins. Protein is composed of, again, the triplet, the CH and the O, and it also contains nitrogen. And in some proteins, there will also be trace of sulfur as well. The building block for proteins, the monomer of protein, is something called amino acid. Amino acid. Right. So the building block for carbohydrates, can someone remind me what do we call the building blocks for carbohydrates? Just try to draw the parallel here for you. Oh dear. You can look back to the notes. What do we call the building blocks for carbohydrates? It's just a single word that I'm looking for. Yes, but what kind of monomer? There's a question. Do we need to know the formulas and drawings of chain of lipids? No, you don't have to draw anything, okay? You don't have to know the formula. No. Okay, so what is the monomer for carbohydrates? Oh no, this is not good.
Is it
Let me
polymonomer?
say that
Or
one more time. I'm sorry. I
a
didn't hear.
polymonomer?
Polymer? Well,
Polymer?
okay, polymer is the end result. Yeah, good try though. Yeah, another.
Is it monosaccharides?
Thank you. Okay, you saved me from despair. It's a monosaccharide. Remember this, guys? Single sugar, right? Right. There are different types of monosaccharides. Right. In fact, we talk about 3 types: glucose, galactose, and fructose. Okay, there are 3 different kinds of right. Now when I mention it, you probably hopefully will ring a bell. Okay, so 3 monosaccharides, single sugar. Okay, that can combine to form more things, right? Okay, so that's the monomer for carbohydrates. The monomer for proteins, they are called amino acids. Amino acids. Right? So instead of just 3 types, there are 20 different amino acids. Okay? 20 amino acids. You don't have to memorize all of them, but you should know there are 20 amino acids. 9 of them are essential. What does essential mean? Do you remember from a couple slides ago? Help me out
So
here.
it means the body cannot make it, so we have to take it from the food or something like this.
Exactly. Right? 9 of them you can't make, and you gotta eat them. So these are the 9 essential amino acids. You don't have to memorize them. Okay. If you're not a picky eater, you should be able to obtain these essential amino acids just through your regular diet. Okay. Some food are particularly high in one form of amino acid than the other. For example, avocado, beans, rice, almond, seaweed, they are particularly rich in something called phenylalanine. Now, they're talking about phenylalanine. There is a genetic condition called PKU. If you are a mother, you would know the first thing they do, well, in Canada anyways, when your baby is born, they would do what's called like a heel prick. Okay, heel prick. They do a little cut at the heel of the baby, and they take a bit of blood and they test for many things. Amongst the various conditions they test for, one of them is PKU, okay, phenylketonuria. In this rare genetic condition, the baby cannot break down phenylalanine, which is one of the essential amino acid. And so if the baby consumes phenylalanine, which would be in baby formula as well as in breast milk, then the phenylalanine is going to cause build-up in the body and in the brain, and it could result in developmental problems. Okay, so they need to test this out, because as soon as they find out if the baby has PKU, they need to be put on a special kind of baby formula that does not have phenylalanine in it. Okay, so that's a little bit of an application for you. Now you might wonder, hey, but I thought phenylalanine is essential. How can they survive without the phenylalanine then, right? So then actually in the formula, they have like almost like an analog, like a substitute for it. So it's not like the actual amino acid, but your body can still use it. So that fixed the problem. All right, I'm gonna draw the amino acid for you. Not that I'm gonna ask you to draw it on the test, but because I want you to understand the different parts of the amino acid. Okay, so here we go. Amino acid. There are 20 different types of amino acid, as I had been telling you. They all have a central carbon in the middle. Central carbon. Okay, and the carbon is connected to 4 things. Okay. It's connected to a hydrogen here. And on one side is connected to C double bond O and then OH. So this here has a name. This is called a carboxyl group, okay, which makes it an acid. Now, if you have been paying attention, we actually saw this earlier in lipids. You don't have to go back, just watch. Okay, see C double bond O and OH, right? Let's go back to here for a second. If you look at the fatty acid, it has the C double bond OH as well. That's why it's called a fatty acid, because this group of molecule makes the molecule an acid. So over here, this carboxyl group, that's what it's called, makes it an acid. On the other side, it has an N and H, 2 H's. This here also has a name. This is called an amino group. So because this molecule has an acid and an amino attached to it, that's why it's called an amino acid. Amino acid. And then on the 4th location, people put an R here. R stands for side chain. Okay, it is different for each amino acid. So there are 20 different side groups. Each amino acid has its own unique side chains. And that is an amino acid right here. Now let's take a look at what happens if I have 2 amino acids. You don't have to draw this next part if you don't want. You can just watch and then you can draw it again later. I'm going to put R1 for side chain number 1. Okay. In fact, don't draw this. Just watch and see if you understand. Okay. So I'm just going to draw it. There we go. That's one amino acid, right? Amino acid number 1. Okay. And like I said, don't draw it. Just watch. I'm going to put another one next to it like that. Okay. But this one would be R number 2. It has a different side chain. I mean, it doesn't have to be different. It could be the same amino acid, but let's just make it different here. Number 2. So how do I connect them together? After all, protein is a polymer, so we are going to be connecting a bunch of amino acids together. What is the name of the process that we use to connect things together? We learned this last class as well.
Is it bonding?
Yep, it's going to form a bond. Very good. But specifically, what's the name of the process?
Like
Covalent.
I'm sorry, what was that?
Covalent bond.
Covalent bond. Yes, we're going to form a covalent bond. But you know what?
No,
Let's go.
it's ionic. It's
dehydration.
ionic. No.
Okay, yeah, thank you. Let's go back to here, right? Look, we wrote it down last time, just so that you know what where we are at. Right? If you have the monomers and you want to connect them together, you have to do dehydration or condensation reaction, right? You need to remove a water molecule before you can form the bond. Does that ring a bell, guys? Hey, you gotta let me know if you don't understand. I can always explain again. So that's what we're gonna do here. We are going to do dehydration synthesis, which means we will remove an OH here and we will remove an H here. So afterwards, again, don't draw this. I just want you to see it so you have an understanding of what's going on. Okay, R1, and then C double bond O. And now I'm going to form a bond. I'm going to form a bond that's going to connect these two things together. Okay, and then plus H2O. The H2O is what came out from the connection. Okay, so this bond that connects the amino acid together, we call it a peptide bond. Okay, peptide bond. And how do you form the peptide bond? You connect a carboxyl group with the amino group. Okay. Now, in case you missed it earlier, like I'm just gonna go back a little bit here, right, to show you it was the same thing we did for the lipids. You remove an H and you remove an OH, right? That's dehydration or condensation reaction. The H and the OH becomes H2O, and you do it 3 times. That's why there are 3 water. Okay, it's a reoccurring theme that you will see. Anytime you want to build a macromolecule, in order to connect things together, you are going to need to remove water. Is that okay? Do you guys have any questions? All right, so here are an example of some, is it 20? Maybe there are 20 here. Okay, so here are the amino acids. And you can see they all have the C and the H, and then the COOH, and then the NH. Don't worry about the negative and the positive. It basically means the same thing for us. It doesn't really matter. Okay. The highlighter part is the side chain. Side chain. So each amino acid has a different side chain. Right? Some of them are just an H, some of them is CH3, some of them have rings. Okay, and depending on what side chain it has, the property of the amino acid is going to be different. Some of them are non-polar, some of them are polar, some are charged, and some are like, positive charge or negatively charged. Right? Now, remember, for amino acid, for proteins, it has CHO and nitrogen and traces of S, sulfur. Right? Of the 20 amino acid, only one of them has sulfur in it. Is this guy right here. Right? So that's where the trace of sulfur comes from. Okay, we did the peptide bond already. Not gonna do it again. Next, we're gonna talk about the 4 levels of protein structure. Okay, 4 levels of protein structure. All proteins have primary, secondary, and tertiary structure, the first 3 levels, and sometimes they have a 4th level. Okay? Not all proteins have the 4th, only some of them do. So let's take a closer look at what it all means. Okay. The primary structure of a protein refers to the unique amino acid sequence in the polypeptide. A polypeptide is just another word for protein, right? Just like polysaccharide is another word for carbohydrates, right? So the protein is made up of amino acids connected end to end. But there are 20 amino acids. So which one do you connect together? Right? It's a little bit different from the case of carbohydrates, right? Carbohydrates in cellulose, in glycogen, and in starch, it's all glucose connected together. So there's no unique sequence. But here we have 20 different choices. It's like having a tray of beads, and I asked you to make a necklace, right? So what colors are you going to link together? Right? Maybe you do the red one, purple one, red one, green one, whatever, right? Like that sequence, that unique amino acid sequence is going to be called the primary structure of your amino acid. Okay? So there are 20 amino acids here. Which one are you going to use to build your protein? Okay? Maybe you use leucine and then methionine and then tyrosine here, and then maybe use another leucine, so on and so forth, right? So if you read out the sequence of amino acid in the protein, that sequence is called the primary structure. And the exact sequence is actually determined by your DNA. We will learn this in lecture 7. Right? You can actually look at the DNA code and figure out exactly what the protein sequence is going to be. Right? So that's the primary structure, the unique amino acid sequence when you read it from one end to another end. The secondary structure refers to whether your protein will form these coils or whether it will form these zigzag pattern. Okay, so here is like a prop that might or might not help you. I don't know. I've been doing using this, and some people find it helpful, some people find it not. So this would be like your protein. Okay, I don't have the beads on them. Just visualize there are beads on them. Okay. So if you read the bead color, like red, blue, green, orange, blue, blue, green, purple, whatever, that sequence is like the amino acid sequence, and that's the primary structure. Now, what happens once you connect all the amino acids together is that some parts are actually going to coil up, and then it will form these little spirals, okay? Form these little spirals. These spirals, they have a name. They are called alpha helix. Right? Sometimes they will have multiple helix. Okay, maybe they will form another helix here. Right? And then the whole thing is made up of alpha helix. right? But other times, instead of forming the helix here, right, they would form these zigzag pattern, okay, zigzag pattern like this. Okay, so just like up and down, zigzag, not spiral, right? So these zigzag, we call them beta leader sheets. Okay, so the secondary structure basically tells you what portion of the protein is the helix and what portion of the protein is the zigzag. Okay? It could be all helices, it could be all sheets, or it could be a combination of both. Right? It depends on the protein. And these alpha helices and beta sheets, they are all stabilized by hydrogen bonds formed between the peptide backbone. Okay, so when you form the peptide bond, it's also able to form these hydrogen bonds, which we talked about last time. You know, one part is slightly positive, one part is slightly negative, and therefore they act as magnets, and they help stabilize these, secondary structure. So in this picture here, the ribbons at the back, the green ribbons, those are the alpha helix. And these arrows, they are used to represent the beta sheets. Does anybody have any question about the difference between primary and secondary? Okay, side story. Has anybody heard of mad cow disease? Is that something you're familiar with? Maybe? Mad cow disease, yeah, some of you heard of it. Okay, so for those of you who don't know, mad cow disease is something you can catch by consuming contaminated beef. Okay, it's a disease from the cow, but you can get it if you eat contaminated beef. Okay, so what is it? Well, in cows and in us, there is a protein called a prion protein. Okay, it's a normal protein that we have. Not really sure what it does, but the normal version of the protein is made up of just alpha helices. Okay, in the secondary structure of the prion protein, it's all alpha helices. For mad cow diseases, okay, people with mad cow disease or cows with mad cow disease, their prion protein is misfolded. So instead of having only alpha helices, they have beta sheets as well. Okay? And what happens is, this is a little bit strange, but once you have a misfolded protein, the misfolded protein will go and find a normal protein, and then it will cause it to become misfolded as well. Okay, so now you have two misfolded proteins, and they will go find more normal protein and change them as well. So it starts off like a chain reaction, and before you know it, all your prion proteins are gonna get converted into misfolded protein. And these misfolded proteins have a tendency to clump together. And when they clump together and form a big blob, it kills the neurons in the brain. This is what the brain looks like for someone with mad cow disease. Okay, the proper name for mad cow disease is spongiform encephalitis. Okay, encephalitis means your brain is inflamed, and spongiform means it looks like a sponge full of holes. Okay, needless to say, it is deadly. Now, the mad cow disease is it's not caused by bacteria or viruses. It's not even caused by living organism. It's caused by misfolded protein. So you can cook your food, you cook your beef, like well done, and eat it, and you might still catch it. Okay, it's not something you can kill by heat because it's not living, right? Okay, so that's why understanding protein structure, it's kind of useful for scientists, right? For people who are studying these things.
I have a question. So what would a normal brain
Look
look like compared to this one here?
Yeah, that's a good question. You know what? I should include a normal picture. Short answer to that is the normal brain would have no white gaps like this. Okay, all these white gaps are basically dead neurons. Okay, once they die, they leave behind a hole. Okay, yeah,
I have a question. Sorry.
sure.
So I'm so sorry. I didn't get in the mid cow disease. What is the problem for the protein? The normal protein I can see, but what happened for the other one?
Okay, so the normal protein only has the alpha helix. Okay, for the mad cow disease, the protein is misfolded. It doesn't just have alpha helices, it also has the beta sheets. And then it causes more normal protein to misfold. That's the problem. Okay. Now, yeah, Cheyenne asks, is there a cure if you get it? I think the mortality rate is pretty high. But you know, if they suspect you have this thing and they catch it early on, I think there might be some ways to prevent the aggregates, like the proteins, from clumping together. But the outlook is not good. All right, so we have primary, we have secondary. Now we have to move on to tertiary. Okay. So I want to show you this picture here on the top right. The top right here just shows you when you connect multiple amino acids together, like what does it look like, right? I tried to draw it for you earlier, but let's take a closer look here. That's one amino acid. That's another amino acid, right here. And then that's a third one right here. And then that's another one, a fourth one right here. So the side chains are going to be different. However, if you look at the backbone, it's the same. It's like NCC, 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 NCC. Like it's the same thing repeated over and over again, right? So that's what it means by the peptide backbone. This backbone can form hydrogen bonds to help stabilize the alpha helices and the beta sheets that I told you about. But when it comes to the tertiary structure, what happens is, like the protein we have here, that, you know, we got the loops and the zigzag. What it will end up doing is it will actually fold up into like a three-dimensional shape. So kind of like a ball shape sometimes oval shape. And this overall three-dimensional shape of the protein is what we call the tertiary structure. Okay. The overall three-dimensional structure is actually stabilized by interaction of the side chains. And remember, there are 20 different kinds of side chains. So side chains that are hydrophobic might tend to come together. Side chains that are positive and negative might form an ionic bond, and so on and so forth. You don't have to memorize these names, okay? But it's important to know that the secondary structure is stabilized by the backbone, but the tertiary structures, the 3D shape, is stabilized by the side chains. Oh, I forgot to give you a break. I'm so sorry. But we are almost there. So, you know, maybe we'll just end early. Okay. Sorry about that. Sometimes I get too into talking about these things and I forgot that you guys need a break. All right.
Secondary structure stabilized by hydrogen bond.
Secondary, that's right. Secondary
Yeah,
structure
yeah.
stabilized. That's right.
Okay.
Very good. Now, some protein cannot function on its own. So, you know, you would have like a green peptide that we make, but then this is not functional on its own. The pink one is not functional on its own either. So what happens is they have to come together, and now it forms a complex. Okay, so together they could perform some function. Right? Not all proteins need to do this, just some. Right? And when they do come together, when they do form the complex, then it has a quaternary structure. Right? Quaternary structure is basically saying, you know, which peptide came together. So for example, in this particular protein, we need two pink ones and two purple ones. That's the quaternary structure. Okay, that's the quaternary structure. In another protein, you might need, you know, two pink, one green, and one blue. That's its quaternary structure. Like which parts do we need to put together to form a functional complex? That's what it means. Now, the protein shape is very important to its function. Okay? The shape of protein actually has a name. We say the conformation of the protein. As you saw with the mad cow, if you misfold the protein, then it could lead to disease. But more importantly, the shape of the protein has what we call like a key and lock relationship with its function. Right? That is, if you change the shape of the key, you can't open the lock anymore. If you change the shape of the protein, it will not function in its normal way anymore. And sometimes it will stop functioning altogether. And either way, it's going to be a bad outcome. So how does the protein change shape? When a protein breaks down, when it unravels, like this is the all fold up shape, right? But there are various conditions that would cause it to open up and unravel itself. Once it's unraveled, we say it is denatured. Denatured. Okay, a denatured protein is a protein that has been unwrapped, unfolded, okay, and it's no longer going to be functional. Factors that can denature protein includes temperature. Right? When you cook an egg, you are basically denaturing the protein. Right? Where do you find the protein in an egg? Do you know? Like is it in the yolk or is it in the white?
Yolk.
White.
It's in the
Yeah,
whites.
the white, yeah, okay. Good try though. The yolk is more like lipid, okay? So when you cook the yolk, it goes from transparent to white. The changing of the color, that actually is the protein denaturing, okay? And it's irreversible. Once you denature it, like you cannot uncook an egg, right? Same thing with the meat. Meat is mostly protein, and when it goes from the pink color to the brown color, it's also protein denaturation. You can also denature the protein by changing the pH, right? Making the surrounding more acidic or basic, changing the salt concentration, or changing the type of solution. Right? If you change a solution from hydrophilic solution to a hydrophobic solution, you will unravel the protein. Now, write this down. When you use things like heat and pH and these things, these will only disrupt secondary, tertiary, and quaternary structure. Okay, the primary structure, for the most part, will be unaffected. Okay, because the covalent bond that holds the amino acid together are really strong. Right? Temperature alone, pH alone, these are not going to be able to break down the structure. Not going to be able to break down the primary structure. Okay.
Are all of these factors irreversible, or is it just the temperature?
Good question. Yes and no, right? Depend how extreme the pH is changed, right? Sometimes if it's just a few degree pH level higher, like you might be able to reverse the effects by returning it to normal pH. But yeah, most of the time, these things in extreme form is irreversible. Okay, I want to give you another example of how changing the amino acid sequence can have significant consequences. We have something called hemoglobin in our red cells. Okay? It's a protein that has 4 polypeptides in it. Okay, hemoglobin is important for transporting oxygen as well as carbon dioxide. So it has 146 amino acid in it. Right, the first amino acid is valine, and then histidine, leucine, threonine. So when you read this sequence all the way to 146, which level of the structure is this? Is it primary, secondary, tertiary, or quaternary? When you read the amino acid sequence,
It's the primary.
it is the primary. And what determines the primary sequence?
Amino acids?
Oh, yeah, sorry. The amino acid is the primary sequence. But who tells you to connect these things together? Like what tells you in the body to connect the valine with the histidine and so on and so forth?
Peptide.
Polypeptide
Good try, not quite. So
by the DNA they are.
DNA, that's right.
DNA.
Thank you. Yes, DNA, right? Your genetic information tells you what to connect together. Okay? Now, your DNA can change, right? We call that mutation. There are many things that could cause mutation, such as exposure to UV light, such as exposure to carcinogens, right? They can all cause mutation. Now, in sickle cell anemia, which is when a person is anemic, they cannot transport oxygen effectively. So they will be tired, they will have shortness of breath, so on and so forth. In sickle cell anemia, the DNA is mutated. So the instruction to make hemoglobin is wrong. And when your body tries to make hemoglobin using the wrong instruction, instead of using GLU, which stands for glutamic acid, your body use valine instead. So of the 146 amino acid, if you change number 6, only number 6, then that's enough to cause sickle cell anemia, right? So in the misfolded hemoglobin, it causes your normal red cell to become these shapes. These are sickle red cells. Okay, they are banana shaped or moon crest shaped, depending how you want to describe it, and they cannot carry oxygen. Okay, they get stuck in your capillary. It causes quite a bit of pain, and that's an example of where if the primary sequence is wrong, it will affect the secondary, tertiary, as well as quaternary structure in this case, causing the protein to misfold and have a different function. Questions about
So
this?
basically, it's like a domino effect.
Yeah. One thing leads to the next, right? And it's usually bad outcome.
Okay.
Yeah. So more on this in lecture 8. Okay, this is just like a foreshadowing, if you would. Okay, finally, what do we use proteins for? Well, proteins are important in many of our bodily structures. Muscle has a lot of proteins in it. Hairs, we don't have horns, but like animal horns, they are proteins. Uh, Nails, spider webs, all these things are made up of protein. Hormones can also be proteins. We learned that sex hormones are steroids, but in addition to sex hormone, there are many other hormones in the body. You probably are familiar with insulin. Okay? Insulin is a hormone that helps regulate blood sugar. That would be a protein hormone. Okay? We can use protein as an energy source, but again, our preferred source of energy is glucose. But when we also have protein, we can burn a little bit of it as well. Proteins are also important because they form enzymes. Okay, and enzymes are needed for chemical reactions in the body. Chemical reactions in the body. We already talked about breaking down lactose needs an enzymes, right? That's just one example. You have hundreds and hundreds of enzymes in your bloodstream carrying many different reactions in the body. And all of these enzymes are basically made up of proteins. Just a random news article here. Some people, poachers, they hunt down rhinos, and then they cut off their horns because in some cultures, people actually buy the rhino horns and then they grind it into powder and then they consume it because they believe the rhino horn has medicinal value. Okay, and some people believe the rhino horn is gonna increase the sex drive. Same thing with the scale of the pangolin. Okay, people believe there are there are like medicinal value to it. For example, They believe it could cure asthma. Right? So they, hunt down these animals and then take the scale and then they grind it in powder and consume it. But I'm here to tell you that these structures, the horns, the scale, they are essentially the same thing as our nails and hairs. They are all made of the actually same protein. Okay. So there's nothing mystical about it. There's nothing special about them. If you're gonna, drink down rhino horn powder, you can grind down your own fingernail powder and drink it. It would be the same effect, right? It sounds kind of gross, right? And that's because it is, right? It doesn't actually have any benefits in doing so. Okay. So people, if they try to understand it a little bit more, if they try to read more about these things, then maybe they wouldn't drive some of these animals to extinction. Anyway, back to enzymes. So again, enzymes are proteins, and the function of proteins is to catalyze. That's a fancy word for speeding up. They try to speed up chemical reaction in a living system. Now, how does it work? Well, if you look at this graph here, the y-axis here is energy, and this is time. And reactants are basically like the ingredients of a reaction, and then the product is like what you get in the end, right? Now, here is the thing. In order for a reaction to take place, you actually need to supply a little bit of energy first, okay? It's, think about going down a ski hill. You have to go up the hill first, right? Okay. So the amount of energy that you supply to start a reaction is called EA, activation energy. Normally, that energy is quite high for a lot of the reaction that takes place in the body. We don't have that energy in the body to get these reaction going. Right? So the purpose of the enzyme is to lower the energy requirement to kickstart a reaction, so that we can perform it at a lower temperature, for example. Okay. Remember last class I told you we burn glucose, right, with oxygen, and then we produce carbon dioxide and water. That type of reaction is the same thing as like burning gasoline, right? Like octane, that's the gasoline that you put in your car, right? And you burn it, and as you know, it gets very hot, right? It just touch the hood of your car after it's been running for a while. You will know how much heat it produces, right? Those kind of heat will kill our cells if we do this kind of reaction in the body. So with the help of the enzyme, we lower the activation energy requirement, and we are able to perform these reactions safely at a lower temperature inside our body. Now, a couple of things here to show you about enzyme. Each enzyme, first of all, just to remind you, they are proteins, and each of the enzyme has a particular shape. Right? If you change the shape, it won't work anymore. And here's why. The enzyme will usually have a pocket. Okay? The pocket is called an active site. Pocket. Okay? These pocket is where you will put the ingredients in for the reaction. And the ingredients are called substrate. I know there are a lot of terms like substrate, reactants, ingredients, they all mean the same thing. Okay? So over here, we have an enzyme, and let's say that this red cube here, let's say that is glucose, and let's say this here is fructose. Okay, so the enzyme will grab onto these substrates, will grab onto these ingredients, and then it will allow the reaction to happen. It will connect them, for example, and it will split out the product. When you take glucose and fructose and you put them together, what's the product? Do you remember?
Sucrose.
Very good. So this is sucrose. Now, here is the reverse. If you want to break this thing down, if you eat a spoonful of sugar and your body wants to break it down, you also need an enzyme to do it. So the enzyme, the active site, will allow the substrate to fit in, it will break it, and then it will split out the product. So you see, the active site is of a particular shape. If you heat up the enzyme, right? Let's say we have a really high fever, right? Then the enzyme might misfold. And instead of having like a square here, it might become like a triangle. And once you change the shape of the active site, once you change the shape of the pocket, the ingredient will no longer fit. So you will not be able to carry out the reaction. That's why prolonged high fever is dangerous. Your enzymes literally will unfold. And once they misfold, that's it, right? Like you cannot fold them back. You will have to wait to make more of it, right? Sources of protein. Meats are excellent source of protein. Chicken breast seems to be the healthier choice compared to pork or beef, because those are red meat, right? And red meat are generally less healthy. For vegetarian and vegans, bean, soy, nuts, they are all rich in proteins. We all need proteins, okay? We cannot have a protein-free diet. You are going to have malnutrition if you don't have enough proteins in your diet. Okay, there are some multiple choice here, but I don't want to spend time on them. You can try them out, and you know, maybe we can take it up later. But I want to finish the lesson with nucleic acid, and that way you can start studying for the quiz, right? Remember, the quiz is next Thursday. Now, here is what I want you to know, okay? I just want to say it so that you know there's no confusion. It is an online course, and I know that we are using the lockdown browser so that you cannot access the files and whatnot. But I encourage you to create like a help sheet for yourself. All right, there are a lot of materials here. If there are some things that you just can't memorize, you can write it down on the help sheet. Right, so not too long, just maybe front and back on the side, so you can refer to it when you are doing the quiz or doing the test. Okay, that's fine. That's not cheating. Okay, like I'm encouraging to do that. Right? What I don't want you to be doing, I mean, it's your choice. If you want to do it, I can't stop you from doing it. But what you should not be doing is have your whole binder, okay, in front of you. Trust me, you're not gonna have time to flip through your notes and find things you don't remember. Okay, when you prepare your help sheet, you know exactly what it is that you can't remember, and so when you see a question about it, then you know where to look for it, right? Okay, so I'm not saying you don't have to study. You still need to study as if this is a in-person test, but have a help sheet next to you. It will make it a little bit easier for you. Okay, I also posted information about the tutor. Okay, we have free tutoring service at Centennial. Please take advantage of it. When I say free, it's not really free. You already pay your tuition for it, right? Okay. I don't know if you saw the announcement. I'm just gonna quickly show you anyways. The person is offering both online and in-person tutoring if you need it. You guys are 202, right? I think. Yeah, so Kian, that's his name. And this is his schedule, and you can book it. There's a link here. You can click in. I think there's a form you need to fill in. But they get filled up really quickly, these tutors, okay? Because they're not just doing biology. You can see they're also doing chemistry and pre-anatomy and physiology. So if you think you need help, if this is overwhelming for you and you need someone to explain it to you again, please book the tutor. He should be able to help you. I am also available for extra help. Okay, email me if you have any question at all. If you need to meet, over Zoom to go through some of the stuff, I am available to do that as well. Okay, I want you to do well on the first quiz, but you have to start studying. Okay? You got to prepare for it. Someone had a question. I saw someone unmute, or it was just my imagination. Okay. We're going to spend the last 20 minutes talking about nucleic acid. It's not too difficult. It should be much easier than proteins. These here in the picture, do you know what you're looking at in the picture here? What are these colorful gummy worm looking stuff?
Chromosomes.
Chromosome.
That's right, they are chromosomes. Do you know why they are called chromosome? Chromo means color. Zome is just body. So these are bodies that suck up colors. That's why they're called chromosomes. How many chromosomes do you have?
21.
Good try. Anyone else?
24.
24.
Also good try. Maybe one more. You have 23 from mom and 23 from dad, which means you have 46, okay? 23 from each parent. 23 from the sperm, 23 from the egg, and then you get 46. Okay. Nucleic acid. It's composed of CHO, the triplet, and then N and P. Basically, the 5 elements that makes up over 96% of the body, right? Now, by now, you should, when you're studying, you should create a table that has all the different macromolecules, and then you should list all the monomers and all the elements in it, right? For example, and all the functions and all the food sources. So for carbohydrates, the monomers are monosaccharide. For proteins, the monomers are amino acid. For nucleic acid, the monomers are something called nucleotide. Nucleotides. Okay, more on that in a bit. Now, thank goodness there are only 2 types of nucleic acids. Right? DNA is what most people are familiar with, but there are also something called RNA. Right? You might have heard of it over the pandemic. The vaccine that you might have received is an mRNA vaccine. So that's a type of nucleic acid. The full name for DNA and RNA are listed here, and you can memorize it yourself. So here is a nucleotide. A nucleotide has 3 parts to it. There is a phosphate, which is the PO4 that we have been talking about, plus a sugar and a nitrogen base. Nitrogen base. So the phosphate group looks like this, PO4, -3. The sugar is a pentagon, and you can see the red sugar here and the blue sugar here. They look exactly the same, except over here it has OH, but over here is missing an oxygen. So this sugar that is missing an oxygen, it's called a deoxyribose. Deoxy means it's missing an oxygen. So this would be a nucleotide for DNA. Deoxyribose nucleic acid. Whereas this one here would be for RNA, ribonucleic acid. The sugar is going to be connected to a nitrogenous base, or you can say nitrogen base if it's easier. It's called nitrogenous base because it contains nitrogen, right? And depending on whether you're looking at DNA or RNA, the kind of base that you can have, that you're allowed to have, is a little bit different. So both of them has A, G, and C in it, A, G, and C. But for RNA, you have U. And in DNA, you have T. So A, G, C are standard for both kinds of nucleic acid. But for DNA, you would have T. And for RNA, you will have U. And if it's confusing right now, we will be using this quite a lot throughout the course. Eventually, it will become second nature. You will know which one is which. Okay. Now, the nitrogenous base can be separated into 2 categories, depending on how many rings it has. Purines have 2 rings in it. Pyrimidine has only a single ring. Okay. So the 2 purines are adenine, which is what the A is, and guanine. Now, this is how you can remember it. You can remember the phrase pure as gold. Okay, remember pure as gold. So purines are adenine and guanine. They are 2 rings. Everything else is pyrimidine, and it only has 1 ring in it. Okay, so cytosine is C, thymine is T only in DNA, and uracil only in RNA. Any questions? Okay, just a couple more slides here. Oh, you have a question? Go ahead.
But
Alistair.
actually, can I ask a question? Just I want to say something because there is lots of information, and so I don't know. I sometimes I get confused. I don't know how can I memorize them.
Yeah, so I know it's a lot of information. So for biology, it's very important for you to write out the information as you're studying them. Reading the notes is good, but reading is not gonna help you remember as much, which is what the study guides are designed for, right? Okay, if you fill out the study guides, you force yourself to write it out. That way, it will help you remember it. Okay, and or also if you organize them in charts and flow charts and concept minds, that tend to help you remember it a little bit better as well. Okay, and also that's why we have the recordings. Sometimes students, if they find if you listen to it one more time, then it's gonna go in a little bit better. All right. I have students who tell me like they listen to the lectures while they're driving or something like that. I'm not telling you that's what you have to do, but some
No,
people find it
actually,
helpful.
I do it. I do it. Yeah,
Yeah. So
I recorded and listened it during the driving. Yeah, thank you.
yeah. Thank you. Thank you. Okay, so we're going to talk about the DNA structure. You probably have seen DNA before in like movies and stuff like that, and it's like a twisted ladder. Okay, twisted ladder. Right? So if you think of like a ladder like this, right? And if you take the ladder and then you twist it, then you basically have DNA. Now listen to me carefully, okay, to help you understand this. The sides of the ladder, right, like here, the side here, that is called the sugar phosphate. Sugar phosphate backbone. It's just alternating sugar and phosphate, sugar and phosphate. Okay. The yellow here is the phosphate, the blue is the sugar. So phosphate, sugar, phosphate, sugar, phosphate, sugar all the way. The rocks that you are climbing on, those are the bases. Okay. I want you to take a closer look at the picture on the right here. Right? You can see T goes with A and C goes with G. And that rule does not change. Okay. Whenever you have T on one side, the other side is gonna be A. Whenever you have A on one side, the other side is going to be T. Now, let me try to ask you, see if you remember. We have 2 kinds of bases, right? We have the purines. Do you remember what the purines are? Which 2 letters are the purines? What's the phrase I asked you to remember, huh?
A and G.
A.
A and G, pure as gold, right? Very good. And how many rings does purines have?
Two.
2. That's right. The way I remember is purine has 2 syllables, so 2 rings. Okay, now pyrimidine, that's 1 ring, okay, which is everything else. So like T and C, right? T and C. I mean U as well, but then there's no U in DNA. So listen, look at the pairing. T is a pyrimidine, so that's 1 ring. A is a purine, so that's 2 rings. So over here across is 3 rings. C is 1 ring, G is 2 rings. So across again is 3 rings. And if you check any of these, it is always 3 rings wide. And that's important. You want the width of the DNA to be the same width along the whole length for stability. Imagine a ladder. If you have a ladder that's like, bulging at some location and then like skinny in the other location, right? Like wider and shorter, the ladder is not stable, right? It's going to wobble all over the place and it might even snap, right? So we don't want that in our DNA, right? That's why there is the pairing rule. A purine will always pair with pyrimidine. Specifically, A always pairs with T, and G always pair with C. Okay, just remember, you can spell the word at. They always pair it up. So that's the pairing rule, right? Meaning, if you know one strand, you will be able to predict what's on the other side. Right? This predictability, this property that allows you to predict what's on the other half of the DNA is called complementary. Complementary. You can predict the other half. The DNA, if you look at it, it's also upside down compared to each other. We call that anti-parallel. Okay, anti-parallel. Okay? We will talk about the significance of that later. But two properties of DNA: complementary, meaning you can predict the other side if you know one side, and anti-parallel. One side is upside down relative to the other. Quickly, if I give you this strand, can you tell me what is the missing half? Okay, just take a moment and do it. You don't have to tell me. You can do it. I'll give you like half a minute or something, and then I'll write out the answer. Think about what pairs with what. Okay, so what base does A pair with? Do you remember? What
T.
does? That's right. A
T.
pairs with T, and G pairs with
C.
C, and that's all you have to know, right? C goes with G, and you can easily come up with the other strand. This property of DNA is called complementary. If you know one half, you can predict the other half. Here is another important question. Suppose a cell has 40% adenine. Adenine is A. Find the percentage of the other three. If you have 40% A, you will also have 40% of what?
T.
That's right, because A pairs with T, right? Anytime you see an A, guarantee you're gonna find a T. I stuck to it. So 40% A, 40% T. Together, this makes 80%. Which means 100 minus 80, you have 20% left. And the 20%,
G.
exactly, the 20% is split between the C and the G, which is 10% and 10%. Okay. Let me do one more with you. Let's say we have 30% C. Right, this is a different question now, okay? If you have 30% C, you must also have 30% of which one?
T.
G. That's right. C plus G gives you 60% in total.
And 40% AT.
That's right. And 100 minus 60 is 40, which means you will
20.
split the 40 into 20 and 20. That's right. That's all. Last slide. RNA is different from DNA because it's actually not double-stranded. It is single-stranded, only one strand. And the backbone is made up of phosphate and the ribose sugar. It has an extra oxygen compared to DNA. And the nitrogen bases include adenine, cytosine, and guanine, but it doesn't have any T. It doesn't have any thymine. Instead, it has uracil. Now, there are different kinds of RNA, which are listed here. You do not have to know the functions. We will learn about that later on in the course. Okay, but just be aware that there are different types of RNA. So that's it. I mean, you should try these questions. We will take it up maybe next class, next week, but you try them first, okay? And yeah, that's it for today. We will start lecture 3 next week, so make sure you print the notes for it. And I'll post the information about the quiz, okay, later today. Okay, thank you. Take care, guys. Bye-bye.
Bye.
Thank you. Bye bye.